Okay, so per some requests, I'm doing a video lecture for this next class, and I know I had mentioned that we were going to do cognitive enhancement, but I changed my mind. I wanted to be able to dedicate a little bit more time to the cognitive enhancement topic, and so we're going to push that back until the final unit of the class, where we'll be doing a lot more discussion. So for this lecture, I thought we would talk about sex differences in psychopharmacology, given that we're at a all-female school. I thought it would be interesting to talk about how differences in gender/sex affect the way that drugs affect the body, and also how that relates to psychiatric disorder. So when we're talking about sex differences, what we're talking about is what's called sexual dimorphism. So dimorphism, basically, di meaning two, morphism meaning shapes or forms. So we're talking about that there are two different forms of something relating to sex. And so what we usually think about when we think about sex differences are the sex hormones, which are estrogen and testosterone. And these aren't exactly dimorphic in the sense that males have more testosterone and females have more estrogen, but that doesn't mean that they're restricted to each sex. So males produce both testosterone and estrogen, females produce both testosterone and estrogen. And regardless, both of these sex hormones are produced in either the testes or the ovaries. So that would be what's dimorphic in terms of what sex organs you have that actually make the hormones. And so different levels of these sex hormones lead to behavioral and physical dimorphisms. So when we talk about the dimorphism, we're talking about differences in behavior and physiology. And that's what the purpose of this little duck picture is. Male ducks have the green head exclusively, so females don't have that. So that would be a form of physiological dimorphism. So when we talk about dimorphic effects or behaviors or physiology, they break down into two categories. The first being what's called organizational effects. And these are called organizational because they occur during gestation and they basically set up the system that's going to persist throughout life. So we all basically start with, quote, female brains, and they only become differentiated when this process of masculinization occurs. So masculinization of the brain occurs because of the Y chromosome that males have, and that Y chromosome is what causes the formation of testes, and the testes are what produce testosterone. And at some point during gestation, there's what's called a testosterone surge. And obviously that can only happen in males because only the males have the Y chromosome. Only the males form the testes. And so when this testosterone surge occurs, testosterone enters the brain. And in the brain, it's actually converted to estrogen by a hormone called aromatase. So it's interesting that it's actually the effects of estrogen and estrogen receptors in the male brain that cause the masculinization of the male brain. So if females make more estrogen than males, why doesn't it exert the same effects at the estrogen receptor in the brain during gestation? Basically, if there's estrogen in females gestating and estrogen in males gestating, then why does the estrogen only in males have the masculinization effect, but not in females? So this occurs basically for two reasons. One, the estrogen surge in females occurs later than the testosterone surge in males. So certain things have already kind of happened and been formed in the brain by the time the estrogen surge occurs in females. And also, and probably more importantly, females have what's called alpha-fetoprotein. And this alpha-fetoprotein binds to estrogen in the bloodstream and prevents it from entering the brain. So during this gestation period, the estrogen in females actually doesn't get into the brain to do what it would, to do the masculinization. So an example of an organizational effect in the brain would be effects that occur in the hypothalamus, or that affect the rate of sex hormone release. And that's obviously reflected in the permanent difference in how estrogen is released versus testosterone. So across 28 days here, levels of estrogen in females cycle basically, whereas levels of testosterone release don't. So that would be considered an organizational effect, something that's set up in the brain during gestation and lasts forever. The other kind of dimorphic effect is what's called an activational effect, and these occur basically after you're born, after puberty, sometimes before puberty, but mostly it's easiest to talk about it after puberty. And these activational effects are susceptible to changes in levels of hormones. So for example, if you give testosterone to a female, she'll exhibit some male behaviors. If you give estrogen to a male, he'll exhibit some female behaviors. And I know that those are loaded terms in terms of like what's a male behavior, what's a female behavior. So at least in rodent studies, we can really break down specific male and female behaviors as it pertains to sexual behavior. So here they're measuring what's called lordosis, and lordosis is a female behavior. And here they're basically rating the level of lordosis for behavioral measurement. So here there's basically no lordosis from the female, and on the opposite spectrum, this is sort of the full expression of that. And basically it's just presenting for mating. And the male behavior is obviously, this more arched position. And so if you actually, one manipulation that will be done in labs is to overectomize a female, which is to basically take out her ovaries so she's not producing estrogen anymore. And if you do that, she no longer exhibits this lordosis behavior. And similarly, if you castrate a male, basically take out the testes, he won't exhibit this mating behavior at all. And then conversely, if you overectomize a female and give her an injection of estrogen, this behavior will return. And oppositely, if you give her an injection of testosterone, she'll actually start to behave more like a male. So these are what would be called activational effects. Differences in levels of sex hormones have different effects on behavior. So how do sex hormones signal? I mean, they're hormones, so they signal in the sort of typical way that hormones signal. So first I want to talk about receptors. So there are basically two different types of hormone receptors. They're the classic hormone receptors, which I've alluded to a number of times before. They live inside the cell. So here's the cell membrane, and these are say androgen receptors, but an androgen is a type of hormone. So these are your hormone receptors, these little blue L's. And your hormone passes through the cell membrane, binds to the receptors inside the cell. Those receptors, they dimerize, they form dimers, which means, again, di is two, so they form two. Two of these come together to form one, and then that travels to the nucleus to have these effects in the nucleus on gene expression. So these classic hormone receptors have what are called genomic effects. So these, by regulating gene expression, they exhibit slow and sort of more long-lasting effects. So classic hormone receptors have genomic effects, and while typically we've talked about these membrane receptors, these cytosolic or intracellular receptors actually they exhibit the same functional components as the membrane receptors. So remember, we talked about the three different components of a receptor, one being the binding region, so that's the region that specifically binds the ligand. Second, sort of the functional efficacy region, so that's the region that determines basically how much effect a ligand has. And three, the effector region. So instead of the hormone receptor having the effect that here in the membrane receptor where it binds to these G proteins, instead its effector is to bind to DNA and have an effect there. So those are the classic hormone receptors, and then there are membrane hormone receptors, which are the same thing as what we've talked about before. They're metabotropic. They have a ligand binding region, a membrane region, and then its effector region. and this kicks off a variety of intracellular signaling effects. And so these will be called non-genomic effects. They tend to happen more quickly than these intracellular effects. And so, there are two different types of sex hormone receptors. There are the estrogen receptors, which bind estrogen or estrogens like estradiol. There are two different types of estrogen receptor alpha and estrogen receptor beta, and obviously these are expressed both in men and women. And then there are the androgen receptors. So, like I said before, testosterone is a type of androgen. So these androgen receptors not only bind testosterone, but other kinds of androgens. And these are also expressed in both men and women. And the sort of dimorphism here is, and the difference between these two types of receptors, aside from what they bind, is that they're expressed in somewhat different locations and in different quantities. So the way that we know sort of where receptors are is through immunohistochemistry, which is something I've kind of talked about before. But I want to explain it a little bit more. So basically, you have your cell tissue. So this would be your brain tissue. The brain tissue expresses a number of different proteins, and you have one protein that you're interested in. So let's say this is your estrogen receptor. So the first thing you do is you put on what's a primary antibody. And so that's sort of where this immuno comes from. So basically we're leveraging how the immune system works to be able to label specific proteins. So this primary antibody is going to be from a different species than your experimental model. So if you're using mice, this antibody will have come out of a goat or a rabbit or a donkey or a horse or something like that. And this particular antibody is going to be developed to specifically attack your protein of interest. So this will be, let's say, an antibody from a goat that specifically sort of attacks the estrogen receptor. So you put your primary antibody on, and it goes and it binds to your protein of interest. Then you wash away all the excess antibody, and then you add a secondary antibody. Now the secondary antibody is going to be from a different species than the primary. So if you used a goat primary, then your secondary is going to be from like a donkey or a horse. And because, antibodies are made to attack intruders, your secondary antibody is going to be developed to specifically attack the species of your primary antibody. And this secondary antibody is going to be attached to a reporter that's going to basically allow you to visualize it. And you visualize it through adding a chemical that interacts with that reporter to create a dark stain. So let me just kind of explain this once more. So here we have our mouse tissue. The mouse tissue expresses different kinds of proteins, and we are interested in looking at the estrogen receptor, which is this green receptor here. So we put on our primary antibody. It's from a goat, and it's made to attack estrogen receptors. So this primary antibody is going to specifically bind to the estrogen receptor and not any other receptor. Next, we add on our secondary antibody, which was made in a horse, and it's specifically made to attack goat. So because the primary is from a goat, this antibody is going to bind to all those goat antibodies. And in addition, this secondary antibody is conjugated to this reporter molecule called HRPO. And then once we've done all this, we add on this chemical that basically causes this HRPO to stain. And so what happens is we get this image of the brain. And so on the left side, there's no signal, but you can see on the right side here, we have all these little dark splotches. And then they look even closer at higher magnifications, and you can see that individual cells are being labeled. And they basically did this using immunohistochemistry. There are other methods such as immunofluorescence, and the difference here is that your secondary antibody is attached to a fluorescent tag. So instead of getting the dark splotchy stain, you get a specific color reaction. So this was stained with a green fluorescent protein, so all the cells that they were interested in are expressing this green fluorescence. And then lastly, there's autoradiography, which is basically an X-ray image. So the X-ray picks up radioactive activity, and so what you do is you apply a ligand specific for your receptor of interest that's also linked to a radioactive isotope. so that when you image it, all those receptors that are binding your radioligand show this dark stain. So we've talked about the receptors. Now let's talk about the hormones themselves. So like I've mentioned before, all hormones start as cholesterol. So here's our cholesterol here, and basically all these different hormones, whether they're expressed or whether they're made, is determined by these different enzymes. So an enzyme here, enzyme here, enzyme here, and basically you'll go as far as those enzymes will take you. And you can see that there's different classes of hormones. There are the estrogens, the androgens, the progesterones, and the glucocorticoids and mineralocorticoids. And so as far as testosterone goes, testosterone's right here, and it's a type of androgen, this purple group. And all of these androgens bind to androgen receptors. Then there's also the estrogens. So estrogen is actually a group of hormones that's actually synthesized from testosterone. So you can see the testosterone's here, and this aromatase turns the testosterone into different kinds of estrogens. So there are three kinds of estrogens: estradiol, estriol, and estrone. Estradiol is usually what we're talking about when we talk about estrogen, as it's the primary female sex hormone during adulthood. But during pregnancy, estriol becomes more important, and after menopause, when estradiol isn't being made anymore, estrone becomes more prominent. And estriol is actually the most abundant estrogen in the body, but it's also the weakest. So lastly, I want to talk about dimorphism and psychiatric disorder. So males tend to exhibit more autism, ADHD, and Parkinson's, whereas females tend to have anxiety disorders, depression, post-traumatic stress disorder. And interestingly, they're more prone to develop Alzheimer's disease after menopause. So that suggests that estradiol, which lowers after menopause, might have some sort of neuroprotective effect in the brain. And obviously, when we're talking about psychiatric disorder, we have to talk about the difference between environmental and biological effects. So are these differences in expression of psychiatric disorder because there's a different biology, or is it because males and females tend to experience different life situations? So that's something to keep in mind. So for your homework, I've assigned you an article to read. I want you to read it twice. The first time, just kind of skim through it a little bit, underline and highlight any terms that you don't understand. Then go and use your textbook or the internet to write or type out definitions for those terms. And then you can either email that to me, or if you choose to just write it out and not type it, I just want to see that you did that in class. And then once you've done that, read the article a second time and make a summary that includes the overall claim of the paper, basically what the paper is, the phenomenon that it wants to characterize, so the key pieces of evidence provided to support that claim. So tell me why they think what they think, and then what are the mechanisms that they think underlie the phenomenon. And that'll be due tomorrow on Tuesday, or whenever you happen to read this or see this lecture on Tuesday. So yeah.